Now, with us this morning is really good. I'm glad that you are here to experience it. Any of you who were here on Monday night and the late night, we talked a little bit about bio. We got into some pretty interesting issues. We're going to go deeper into them now. Andrew Hessel of Autodesk is going to moderate. So, Andrew, please do so.
Hello, everyone. So Life 2.0, we're still getting the hang of Life 1.0 in many ways.
Finally figuring out somewhat how it works, I think, yeah.
We're still trying to figure out we might be getting some peeks under the hood, but we're still trying to figure out how to apply it in the real world in many ways. We've heard a lot about health and really some of the challenges that we're dealing with right now. Now, you two are friends, and you both come from very different perspectives in life. You've had a long life touching on many different technologies, still involved in genetics more and more. Ari, not so long a life. I met you in a hacker lab in Sunnyvale, one of the first garage labs I ever saw, up and running. And really starting to bring a whole new generation into the world of life science. So let's start by just the question, Life 2.0, what is it to you? What do you see it as?
For me, it's Life 1.1, in the sense that the main thing I'm focused on these days is trying to bring back extinct species, starting with the woolly mammoth and the passenger pigeon and moving on to the range of extinct species that we might welcome back. I do this as a conservationist in the sense that our organization is called Revive and Restore. We want to bring back the woolly mammoth, not just to have it in zoos, but to have it back in the hundreds of thousands in the north, turning tundra back into mammoth steppe grasslands the way it did before. And likewise, the passenger pigeon, please darken the sun again. There used to be 5 billion of them. We should get at least 1 billion back.
That's, now, and are people taking this seriously?
The people taking it seriously are people who've looked at the technology and realized that there's a lot of very difficult stages, but there are instances of each one of the difficult stages that have already played out. So animals where you have fossils less than about 200,000 years old, there's recoverable, what's called ancient DNA in there. It's also in the museum specimens. And thanks to shotgun sequencing, you can take that very fragmented, very contaminated DNA and having to do pretty high computer muscle on it, much harder than working with living DNA. Nevertheless, you can rebuild the entire genome of this extinct species. And then the idea using George Church's technique and new techniques like CRISPR, which is a genome editing technique, take the closest living relative of, say, the woolly mammoth as the Asian elephant, and basically swap the woolly mammoth genes into the Asian elephant genome, implant that egg in a surprised mother Asian elephant, and out of that hopefully get woolly mammoths who then make more woolly mammoths. Simple.
It's absolutely simple, straightforward. I can see it happening in bedrooms already. The biggest bottleneck, what do you see it being?
The biggest block will be...
Most scientists say funding.
Well, it's funding, but I think that once people catch on that, I mean, pretty soon, the way people say, well, if we can land a man on the moon, we can do so-and-so. Well, if we can bring back the woolly mammoth, we can do so-and-so. And I think some folks are going to say, how much will that cost? What fraction of getting a man on the moon? Oh, 1,000th.
Yeah.
$5 million, something like that. Get woolly mammoths back on the planet, passenger pigeons back on the planet. Somebody's going to say, I want that to happen. And then you're off and running, and people ask how much does it cost, and George Church, whose book I recommend, by the way, Regenesis, his flipped version is to get the woolly mammoth back in 3 years, $5 million, in 5 years, $3 million. Because the technology is moving so rapidly, as people keep saying, 4 to 6 times faster than Moore's law. The costs come down, the sophistication goes up, the capabilities move from impossible to expensive to routine, in just a couple of years. And I expect that bringing back extinct species seems kind of radical now. In a few years, it'll seem like a really conservative thing to be doing, which is exactly what we want.
Ari, you've been a big part of really a wave of democratization in life science. From this garage lab that I saw met you in a number of years ago, you went on to found BioCurious, one of the first community laboratories in life science. Can you share a little bit about what you learned with BioCurious, what you're still learning perhaps?
Sure, and I think it serves us to talk about why we were in a garage. So it's nothing about being in a garage in Silicon Valley, although that can always work to your benefit. It was because we couldn't afford lab space anywhere else. And that shows us a huge problem in the life sciences. As Stuart said, things are getting incredibly cheap, accessible, and people are more and more interested in being an active participant in their own health or even in manipulating cells. And that's what we had noticed because we were those same people. And I was working with people who were sick of academia, sick of not being able to take a risk because the system wouldn't allow for it. You're dependent on your PI, your principal investigator, or where the money is to even try an idea. So the low-hanging fruit is people who think outside the box, what is low-hanging fruit, what's cheap, what's doable, and basically what can we do without anybody else. So we were trying one of those concepts in our garage lab. It was simply to quantify the cancer-killing ability of certain cells. And we ended up being able to do that as a two-person operation. And me having no background in bio kept trying to bring people who knew more and more than I did around me. And what we would do is have some kind of scientist like Rob Carlson, like you. I remember asking you several times if you would come and speak. And people would show up. We completely basically broke the walls on our house because people demanded it. And we ended up needing bigger and bigger space just to convene. People would show up, drive for literally 2 hours from Sacramento just to hear a scientist talk and then to hang out with other people who were interested in the same topic as them, but outside of the walls of academia or institutions.
What kind of backgrounds were the people coming with? Were they from biology or genetic stuff?
Sometimes. And it didn't always matter to them. We have a lot of artists and a lot of musicians. It's no surprise. So there's a DIYbio.org mailing list, which has done a tremendous job at getting people involved. They have thousands of people across the world who share pretty high-level talks about biotech and how you can do it from home to a community lab. And at one point, 2 years ago, co-founder Jason Bobe said 50% were artists. And that it's very interesting when the people who are shaping a revolution are artists.
Creatives.
They look at the world in a different way. And some of these artists are turning into biologists. They're getting the skills that they need in order to do the work that they think needs to be done. And it is possible in a garage and now in community labs. And with all the people coming, we realized we just needed something more. This isn't a two-person operation. This is dozens around us, thousands of people across the country, and it's growing. It's grown tremendously since those few years ago.
How many members does BioCurious have now?
At our lab at any given time, we'll have from 10 to 40 people.
Okay.
We can't fit much more, and that's unfortunate. That's because it was 6 friends who started BioCurious as a lab. So now it's a separate lab that anyone can be a member of in Sunnyvale. And we went on Kickstarter as one of the first projects, thinking that we're going to try to raise $30,000 and we're never going to get it. Today, it's a world of difference with crowdfunding. We ended up raising $35,000, which is a ridiculously low amount that we thought was a lot. And that's a sort of naivete that helped us kind of get the job done. We just didn't give up until it was created. And we see the same phenomenon. Like the person who started our bioluminescent project, which has some seeds in the Glowing Plant Project, which just raised $500,000 on Kickstarter, was started by an artist.
So for those that don't know this, another Kickstarter, this time to make a glowing plant, raised about $500,000 in about 4 or 5 weeks. and has become one of the real lightning rods, so to speak, in this next generation of biotechnology, because they actually intend to send out the seeds for glowing plants. As I understand, they're moving along very well and progressing. Stuart, how are you funding your work with de-extinction? How are you attracting scientists and community members to this project?
The conservation guys don't want us to take any money from conservation sources. And so
You're threatening their domain.
And they needn't worry, because who's showing up is, we have these founding funders putting in $10,000 apiece, and there's 15 of them so far, and probably more to come. They tend to be VCs and people from digital tech, and folks who I think want to have a foot in what they see as the next platform. Plus just being part of something wild and crazy and charismatic, and yet actually potentially quite practical, bringing back extinct species. It feels like they know it's going to be a century-long story. It's one of the reasons we're doing it within the Long Now Foundation. Woolly mammoths, you get a baby girl woolly mammoth, and she's not going to have a baby girl woolly mammoth for 20 years. Unless we develop artificial uteruses, which maybe that'll occur, but probably not. Or maybe it will. So you're engaging the nature I care about, which is conservation, which is biodiversity and the richness of natural biology, at its time frame and at its level, sort of an evolutionary frame. And that's what I studied at Stanford back in the 50s, was ecology and evolution. What I love about this, and what I think some of our funders are enjoying, is that this is a way to catch up on what's going on with molecular biology. So the stuff you guys are already engaged in, I'm still learning, what that CRISPR means. I wrote it down so everybody can know. This new genome editing technique refers to clustered, regularly interspaced short palindromic repeats. And you're using an enzyme called Cas9. This is getting seriously sophisticated out there, folks. And I think one of the things to understand is the digital code is pretty difficult and complicated. It is so easy and Minecrafty compared to genetic code, which is 3.8 billion years of spaghetti code with no engineering involved at any stage. So reverse engineering it is out of the question. This is very interesting code to work with.
But you make a great point. Like many people are speaking about biotechnology, of DNA code as really the next IT industry, the next programming industry. Is this fair? Particularly, you were just at the Homebrew Computer Club meeting on Monday.
Which I missed. Thank you
Which
for
Stuart
going.
missed. Of course, we know some of the history around this, the professional enthusiasts coming into the field just with interest, enough of an infrastructure foundation to really support those, the first creators and builders in the field, and of course, world-changing results. Do you see, with your recent experience, your long experience, analogies here? Is this fair?
Yeah, so you saw the personal computer guys from that many years ago, cackling together and reminiscing. Do you think your crowd is going to be cackling together and reminiscing like that this many years from now?
I was trying to think about it. I think if that's what we wanted, then we could create that. And short answer, I don't know, but Daniel Cockey, who calls himself Apple employee #1 in the garage, said, in 30 years, you're going to be having one of these. And my instant reaction was, since we were in the Computer History Museum, which was founded by members of the Computer History Homebrew Computer Club, we're going to need some sort of biological history museum. We're going to need to understand that in context.
Oh, I love the idea of starting that right away before there's much history to put in it.
Isn't there?
There you go. Add that to your
You're creating
repertoire.
the history again.
Yeah, and in fact, even in this recent wave, what I look at as the modern age of biotechnology, where it's going digital, really starting with the sequencing projects, the Human Genome Project. Now with being able to 3D print the DNA molecule, we actually have almost a 25-year history just in digital biology. So maybe it is time to start considering that.
And it's well to remember that Homebrew Computer Club came after a whole lot of computation, and you had the long-haired programmers, the hackers of the 70s, basically, and then the narrow tide programmer engineers who had been working for 25 years at that point and had already done mainframes, or had already done mini computers. And along came personal computers, and they were sort of risible to the narrow tide crowd. You know, why would you guys waste your time with these toys? And the answer was because we can do whatever we want with them. And then it turned out that I think got smaller and smaller, and then you're into smartphones and got Google Glass and whatever's next. So this shrinking down of the empowering of amateurs, basically, is I think the stage that we're looking at here. But it's still the case that for us to bring back extinct species, we're not going to do it at BioCurious or DIY Bio. We're going to probably do it at the Wyss Institute at Harvard with George Church's multiplex automated genome engineering machine, which is this robot that replaces squadrons of postdocs with pipettes and generates a whole bunch of writes genes and then generates them into now microbial, but soon many kinds of vertebrates, including the ones we're interested in, generates multiple genes at once in there, which obviously we need to do if we need to move dozens, scores, hundreds of genes from one species to another. So that's all pretty industrial level stuff, but the costs are coming down, and there's going to be glowing plants and stuff like that out there drawing fire away from us. Thank you very much. All those nice people, they're doing a nice safe thing like bringing back woolly mammoths and saber-toothed tigers. Don't worry about them. It's these glowing plant people that we have to worry about.
No, that's not true. Worry about the woolly mammoths.
So what are your thoughts on that, though? Are you really are these DIY researchers? Are these the younger generation coming in? Are they the ones that are going to be doing the projects that catch people's attention more than something as big as being able to bring back extinct species?
As soon as Stuart mentioned that he would be working in George Church's lab, I thought as soon as people see a video of this, they're going to think about, well, how can I recreate George Church's machine?
Bingo.
And what can I do with that? And how can I make it open source and hackable and for the people and not for George Church's lab?
And there's a lot of transparency. George is in academia. He started no end of businesses. He's part of no end of businesses, but his whole approach is transparency. Our whole approach is transparency. And there's an open source aspect to a lot of this end of what is possible with genomics. And so, ideally you'll get some of the benefits of open source, and then there'll be lots of people who, patent something and make a lot of money with it.
So you think we'll see a generation of bio-entrepreneurs coming into the space as we saw with computing? Is there I'm hesitant to use the word killer app, but is there a
resurrection app.
Low-hanging, is there low-hanging fruit in this space that you see that you might think what would you buy? Put it this way, what would you make or buy in this space to start turning it into?
Yeah, you guys are Institute for the Future. Presumably everybody's watching you to tell where the new business breakthroughs are, right? What do you see?
Sure. Next year, our research is going to be around man and machine. So where are we headed with the future of being human, and what does that mean? There's this big issue of identity there when we're realizing what we're made up of microbially. Ratio of 10 to 1,
Right.
non-human cells to human cells.
Right.
And we're just now able to afford to sequence what's living inside and on our bodies.
And in fact, just to interject, a company called uBiome, just another crowd-funded project, just launched, started shipping their kits a few weeks ago to be able to assay microbes anywhere you want to explore them, pretty much on your body, for about $79 per test.
I bought a couple.
Yeah, yeah, really quite impressive. So again, waking people up to the idea that there's a lot of life around them that they're not paying attention to today.
Did you know that there are amateurs out there doing experimental fecal transplants?
They're experimental fecal transplants.
Why not?
Fecal transplants, by the way, if people have gastrointestinal disease, it's usually because you have a population of microbes in your gut that just for whatever reason don't work for you. And fecal transplants have produced some miracle cures, but I didn't know that there was starting to be amateur work done in the space.
You know,
But then
yeah, there is, of course. I mean, it is the simplest medical procedure you can think of. You know, bend over. I mean, it's
Where do you report the results?
amongst probably a small and interesting cadre of people.
We're talking really low-hanging fruit here.
Business idea.
Whoa!
Celebrity feces.
Celebrity feces. So I've seen people take microbes, make cheese out of it, from different parts of the body. Do you see like Newman's Own, in different ways there, food products?
Easy's navel cheese. Wow. Okay. There are already people talking about can we sell mammoth hide and mammoth meat before you got mammoths, and I suppose something that might occur.
And I was recently in China and got to visit the Beijing Genomics Institute, where they showed me, at this point, it was a dead and taxidermic pig that they had made to glow. So they had made several fetal pigs glow. So it's like these glowing plants, but this is taking it a step further to mammals. Mm-hmm. And they were fine, normal pigs, but they glowed when you shined a black light on them. This would never be allowed in the US. There is, at least at one point, there were people who were creating glow-in-the-dark fish, and these are outlawed in the state of California.
Right.
And it's not that difficult, but our reaction is, it's just that. It's very reactionary because we don't have the standards to allow, or regulations that are aware of these new capabilities of science and genetics.
I think some people aren't aware that you can't even use a 23andMe kit in every state just to assay your own
I didn't know
genome
that.
profiles.
On account of creativity issues or what?
I'm not really sure. They need a physician as an intermediate.
It's not in the Bible, so we don't have it in Arkansas. I don't know, anyway.
I don't know.
Were there, in China, were there amateur biotech people emerging? Because in a way, what we're hoping for with nuclear, we're hoping for with GMOs, we're hoping for with a lot of things is that bolder, less overregulated countries are going to be the places where a lot of the breakthroughs happen. And did you see at BGI a sense that they were going to be able to move faster and stranger than, say, we can?
Yes, absolutely yes. They're moving at an incredible pace. And their first feat was to just say that they were going to sequence 0.5% of the human genome. And that was before they even had a facility or any funding. And so retroactively, the Chinese government gave them money because this claim of theirs got written up into Nature. And that's a really big deal.
Right.
And they've been doing things like that ever since, but because of the lowered price cost there, and the ability to do things that just wouldn't pass our regulations here, they're going to move beyond us. And they've already progressed so quickly. They're a major player. And when I was there, after showing me some of the new plants that they've grown, some new hybrids that are meant to be bigger, they grow more quickly and they're more nutritious. They showed me also grouper fish that had their genes spliced, so they were bigger and more delicious. They even make their own yogurt with bacteria, their own special brand of bacteria. And they said they want to be the new Monsanto.
Sort of probiotic bacteria that's even more wonderful than our probiotic bacteria.
Of course, more wonderful.
Yeah.
So,
This is actually fascinating because this is not a case of Chinese copying the U.S. In fact, they're using the U.S.
Creation.
technologies, aluminum machines, et cetera. I've been to BGI myself, and actually being able to move forward because of will, because of scientific training. They have, when I was there were 4,000 bioinformatics scientists, average age 22, working in the facility, which is remarkable. All trained in computers on the way in the door. And then perhaps being more fearless in the exploration of the space.
is it mainly China? Do you see this in India, Philippines, Southeast Asia, other places, or do we know?
I don't have a strong background in or global knowledge of genetics, but I see BGI again and again, and it doesn't strike me that any other country has the capabilities that they do in this arena. You should visit.
That's fantastic. I have one, maybe this is a good point. If you were invited to the White House tomorrow, what's one thing you'd really want them to understand? And then we'll open it up to some questions. But like, what does our government need to pay attention to?
You know, the White House has been wonderful on synthetic biology. They had, right after Craig Venter did a computer-generated genome and planted it into a different creature, and it became the creature of the genome. As he said, the software created its own hardware. They had a great gathering at the White House to basically hold forth about the regulatory environment and the way to think about synthetic biology and where it was headed. And basically, it was a pretty libertarian document they came up with, that this is an area which is moving quickly. It's important that it move quickly. The government should not stand in the way. The government should help be sure that there's no bioterror issues or things like that, which it turns out is in some ways easier to do than people first think. And that let the marketplace decide where the great usefulness is going to be here. So I think that if Obama or any of his people were in the audience here, they'd be nodding away with other people who are nodding and wondering what the people who are not nodding are having a problem about.
Harry, anything to add?
Sure. So there's radical shifts in the way that we're learning today. Some of us talk about how academia is broken and the university will no longer exist in the next 20 years. And I think we need to reflect all of the changes that come out of a changing way of learning. And as it relates to biotech, people are learning outside of academia. I didn't realize I wanted to work in bio until after I graduated college, having been so grateful that I was leaving school. Yeah. So I tried really hard to explore the alternatives for me because I wasn't going to give up on what really mattered. So human health, finding cures, it really matters. And we're not getting to the place that we need to be quickly enough. And there are people like me who want to participate. And some of these people show tremendous promise and brilliance. And I've been so amazed at the people that I've met, just trying to learn a bit, but they're not getting the resources that they need. I don't want to feel like the typical academic who says, well, we need more funding in order to explore this problem a little further. It's really the case that the people who are game changers are the ones that we're not aware of and that aren't pursuing things in traditional ways. So if I were at the White House or just being right here, I think that should be recognized. And if policy-wise, regulation-wise, we can do something to enable the individual who doesn't want to be at a university or at Genentech, then let's do that. Let's create some of these regulations and policies.
Let's take a couple of questions while we still have a few minutes. There's a question just behind here.
Jerry Spiegel from New York. Boy, you just scared the living daylights out of me. And I am fully aware, as most of the people here are, that these sorts of biological experiments have been going on. But the idea that they're going on around the world in places that are completely unregulated really scare me to death. I mean, we've seen what's happened with natural species being introduced into our environment and how they've taken over and destroyed it. What about these artificial things? How do we deal with the fact that this is going on in a completely unregulated way?
That's a common, thank you for asking the question because it's a really common one. And a gene from a different place is not at all like a species from a different place. And even most invasive species are not a problem. They are a problem in a few delicate kinds of places like ocean islands. that are basically closed systems, and some freshwater bodies that are closed systems. Genes have been swapped for 3.8 billion years, and you're in my body at this moment. By the hour, genes are being swapped amongst all of our microbes. That's what they do for a living. You can't even really identify separate species. They call them functional species because they swap their genomes all the time. And so genome swapping is the norm. We're still catching on to that. In terms of actual hazard, the Asilomar conference that was held in 1975, when the capability of what was then called recombinant DNA, gene splicing, first came along, and there was a weird panic reaction from political bodies like city councils. And so the scientists got together and said, what are the actual hazards here? What kind of regulations do we need to generate, what kind of hoods, bunny suits, various things like that, to make sure that there's no real hazards coming out of this? And they came up with a set, and the federal government stepped in and helped regulate those things. And then they dialed the regulations back year after year, decade after decade, as they discovered that there was great safety. Since 1975, exactly no one has been harmed by anything that has gone on in these wet labs. We've had GMO foods since 1996 with alien genes poked into the corn and into the cotton and into the soybeans and the sugar and whatnot. Exactly no health harm or environmental harm has happened from any of that. There's various things that come along and you look at, the scientist says so-and-so, it turns out if they're an advocate scientist or they got something wrong, and then it's the usual outlier science that doesn't hold up. So we've now got a lot of experience of, are there bad things that are potentially happening from this kind of screwing around with genomes? And the answer is not yet. Craig Vander's answer is the one time that biotech was really, really dangerous was when it was sequestered away in bioweapons labs in the Soviet Union and in the United States. Since then, it's been getting safer and safer and safer.
Yeah, right here. Sorry, we are out of time, so we'll have to move it fairly quickly.
All right, Todd Cochran, Geek News Central. The GMO folks are just going to go absolutely crazy over this, the folks that really oppose GMO, and I kind
They've
of
been
laugh.
strangely silent.
It's interesting if they are, but I laugh because we've all been eating GMO foods for a long time.
I've had Et cetera come after. I was at a presentation I gave in England, and they said mammoths are bringing back mammoths is a mammoth distraction, which means they're not going to focus.
But if they hear about manipulation of fish and plants, as soon as that becomes mainstream, people are just going to freak out. And I think
they
your
will
days are
lose the
numbered
young
before they
generation of
start hammering the
environmentalists.
legislation.
Yeah.
Well, if we look back at food history, a lot of commonly held beliefs about food were due to government, so top-down messaging or interesting results from studies that the media thought were great to write about. And to this day, we have no idea what is good nutrition, honestly. I recommend the book, Good Calories, Bad Calories, by Tom. It's an excellent read, and it's not a nutrition book. It presents the real research about why we eat en masse certain categories of food, why we're high carb, why we're low carb, and the answers are very surprising. And when we think about people freaking out, I say it's really going to depend on what the messaging is. It's going to convince us one way or the other, but I think what we're trying to say is you're capable of doing the research yourself. That's what we're trying to do. And the reason that Asilomar worked and the grassroots effort to regulate bio is working is because people are observant, they really care, and what affects everybody else affects them too. And if you're ever interested in hanging out with some people working on this, I know at BioCurious you're going to find people who are going to welcome you, be happy to talk to you about everything that they're doing. We're totally open and transparent, and I'm sure Stuart and his crew are
So perhaps to
going
close on that point, this is an open biotechnology.
Yeah.
It's an open biotechnology that's global. All the information is out there, as opposed to some of the closed secrets, whether it was done by governments or corporations. And we're seeing a new wave of people coming in. I remember with the iGEM program that you mentioned the other night, I would see parents who had been against, just kind of had grown up against genetic modification, seeing their kids learning this stuff, just as 30 years ago you might have learned computers. And they were absolutely enthralled. They said, I've never seen my children so excited about a technology, so engaged, and learning by doing. And so, certainly we're not going to solve, every issue here.
And the important point to add is that there's FBI guys at these iGEM jamborees, and it says weapons of mass destruction on their name tag.
Weapons of
Card,
mass
they
destruction.
hand out the card to everybody with a big smile saying, you guys, you people, are the sensors in this world of potential bio issues. You know what's going on. You know if anybody's particularly either wild and crazy or criminal intent or whatever it is, let us know. Here's our phone number. This is the opposite of what they did back in the hacker days of computers and homebrew.
They call it the
where they
new
would,
FBI.
put hackers in jail. They don't put biohackers in jail anymore. They ask them to help.
And let's close it out on that. Thank you very much for your attention.
Thank you. I got to say, just for anybody who's watching online and wants to pursue this further, the session that Stuart was on Monday night is a great corollary to this one, and Andrew contributed to that. Thank you to all three of you. Really interesting discussion.